Hello. This is the biochemistry final for Stephanie, Dayton, Tyler, and I. Now, we were assigned lipid biosynthesis, and we chose specifically to look at cholesterol biosynthesis. Now, we always think of cholesterol as coming from our diet. We think of heart disease, but of course, as we all know from cell biology, it's very important in the cellular membrane, in the creation of steroids, the synthesis of bile acids, and vitamin D. And in fact, 70% of all the cholesterol in the body that is actually used comes from the cells themselves and is mostly synthesized with the help of the endoplasmic reticulum, usually the smooth. And when we look at cholesterol, we see that it's a 27 carbon structure. It's very complicated, but you would think it would come from multiple intermediates, but in fact, all of the carbon comes from acetyl-CoA, which is produced by glycolysis. And when we look at the structure of cholesterol, what we see is that there are four rings: A, B, C, and D. We see that there's an 8-carbon tail on ring D. There is an OH group on ring A, and there's a double bond on ring B. And this gives it a highly hydrophobic character, though it does have a hydrophilic OH on it, and this gives it all of its very unique properties. And the process of creating cholesterol is an endergonic process which needs 36 moles of ATP, 16 NADPH, and 18 A-CoA to make a single cholesterol. Lots of components are coming together to make this, and this is what we will go over, looking at the energetics, looking at the structure, the chemistry of each of the four steps of cholesterol synthesis. So the first step in this process is the joining together of the acetyl groups from 3 A-CoA and then reducing them to form a mevalonate. And the first two groups are joined together by acetyl-CoA acetyltransferase. And in this, a catalytically active cysteine nucleophilically attacks the CoA, knocking it off, at which point it is joined to another A-CoA to form acetoacetyl-CoA. Now, in order to join the third acetyl group to this, you need HMG-CoA synthase, HMG here standing for beta-hydroxy-beta-methylglutarate. CoA HMG is, of course, much simpler, and this works in the same way. A cysteine group attacks, and then the acetyl group is joined to the other two, but at this point it becomes a carboxylic acid, and it becomes an anion, giving it a polar character, meaning that it's water soluble, though this property will be lost when it becomes a full cholesterol. And in order to finally take these three acetyl groups and remove the final CoA and reduce it, get rid of all the double bonds, make a mevalonate, you need HMG-CoA reductase, and in order to reduce, it needs NADPH. And how this works, it's still being studied, but it seems like an active lysine will attack the CoA, knock it off, at which point it will also bring in the NADPH and transfer electrons to the HMG and reduce it into forming a mevalonate, which again would be a carboxylic acid, an anion, very water soluble. And this is actually this sub-step of step 1 is the rate limiting step for the whole of cholesterol synthesis. And this is done because HMG-CoA reductase, which makes mevalonate, is very, very tightly controlled by the cell itself. In fact, it is actually not in the cytosol. It is bound constitutively to the actual membrane of the endoplasmic reticulum. And the cell uses it to limit cholesterol synthesis because HMG-CoA reductase has another domain which binds cholesterol, and this reduces its efficiency at making new cholesterol. So when cholesterol is in the cell, HMG-CoA activity will be reduced. And this is the target of statins. Statins, the cholesterol-reducing drugs, they will bind to that same active site and stop it, and stop the action of HMG-CoA reductase, and I find that very interesting. And of course, mevalonate will go on to become an isoprene, then a squalane, and finally the cholesterol.
Methylonate is converted into two activated isoprenes. The first three sub-steps of this process occur when a phosphate group is transferred from an ATP molecule to methylonate. Each of these sub-steps has a different enzyme that's used. The enzyme that converts methylonate to 5-phosphomethylonate is methylonate 5-phosphotransferase, and it will add the phosphate group to the fifth carbon. 5-phosphomethylate is then converted into 5-pyrophosphomethylate by the enzyme phosphomethylate kinase. This will then add a second phosphate group on the end, leaving the main carbon chain with 2 phosphate groups attached on the end. 5-pyrophosphomethylate is then converted into 3-phospho-5-pyrophosphomethylate by the enzyme pyrophosphate methylate decarboxylase. During this reaction, the phosphate group will be added to the third carbon. The first 3 substeps of this process convert methylate to 3-phospho-5-pyrophosphomethylate using 3 ATP molecules and 3 different enzymes. The intermediate step produced has a carboxyl group on the end and then a chain of 4 carbons. The third carbon has a methyl and a phosphate group attached to it, and there are 2 phosphate groups on the end of the chain. During the next step, the carboxyl group and the phosphate group from the third carbon will leave using the pyrophosphomethylate decarboxylase enzyme. This step will release carbon dioxide and an inorganic phosphate. The 5-carbon product is released, and it has a double bond between the first and second carbons, and this is known as delta 3 isopentenyl pyrophosphatate, and this is the first of the 2 activated isoprenes. This can then undergo isomerization using isopentenyl pyrophosphate isomerase to produce the second activated isoprene, dimethylallyl pyrophosphate. The isomer is a monomer with alternating alpha helices and beta sheets. The active site is deep within the enzyme, and it consists of a glutamic acid residue, which is highlighted in blue, and a cysteine residue, which is in red. One of the activated isoprenes will bind to this active site, and the 2 residues will interact with it from opposite sides to produce the other activated isoprene. The 2 activated isoprenes produced in this step will then move on to the third step to form squalene.
All right, the third stage of cholesterol biosynthesis occurs within the cytosol and involves the polymerization of 6 5-carbon isoprenes to form a 30-carbon squalene. Picking up where we left off at the last stage, we have 2 isomers. We have dimethylallyl pyrophosphate DMAP and 3,3-isopentenyl pyrophosphate denoted as IPP. These undergo a head-to-tail condensation reaction utilizing or catalyzed by prenyl transferase. Now, prenyl transferase, it comes from a class of enzymes that are responsible for transferring allele groups from one molecule to another. The specific enzyme utilized in this reaction is dimethylallyl transferase, and this allows for IPP to act as a nucleophile attacking the electrophilic DMAP, displacing the diphosphate group right here. Now, head to tail, head just refers to the diphosphate group, and tail is that carbon chain. But that reaction ultimately forms GPP, which is a 10-carbon intermediate, guanylyl pyrophosphate. It's important to note that the negatively charged diphosphate groups found on these intermediates and molecules are responsible for the polarity of the molecule and allow for them to be soluble in aqueous solution and to ultimately undergo catalytic reactions within the cytosol. And that's why that is important. The next reaction is the exact same to the first. It is a head-to-tail reaction utilizing the exact same enzyme. The only difference is IPP is attacking GPP instead of DMAP. Again, it's displacing another diphosphate group. And this now forms a 15-carbon farnesyl pyrophosphate intermediate. Now, the final reaction is a 2-step reaction utilizing 2 FPPs. And to know where we got that second FPP from, I just have a second pathway, the exact same, next to it, just to show if anybody was curious where that second FPP came from. And I'm going to now scroll down a little bit to show this reaction a little bit more in depth. So this is the final, basically, step in this stage. And it involves 2 farnesyl pyrophosphate intermediates going into it like a head-to-head condensation reaction with and utilizing squalene synthase. Now, in the top right corner here, we have a picture of squalene synthase And according to the Journal of Biochem, this enzyme is entirely alpha helical, in which is aligned and arranged in 3 layers. The protein is folded as a single domain with a single channel running through the center. And I believe this is that single channel or cleft running through the center of the protein. And this step undergoes 2 half reactions in which both FPP bind to one end of the channel that is exposed to the solvent, a little bit lower on the enzyme, and undergo that first half reaction. So the first half reaction involves them, again, going head to head, condensation reaction, displacing a diphosphate group and forming a pre-squalene pyrophosphate, which is very stable and is still water soluble. Now, this stable pyrophosphate intermediate moves into the deep pocket. So it's going to move kind of up into this deep pocket where the flap is and undergo that second half reaction. This is where it gets reduced again by NADPH, forming NADP, plus displacing its last diphosphate group and forming a nonpolar now squalene. So that last step is very important because it's the first committed step in the formation of cholesterol. It's unable to go in a different direction once it goes through that pathway. And kind of looking back at acetyl-CoA, this pathway requires 18 ATP molecules to produce one squalene. So it's a very endergonic pathway. And I just want to scroll down real quick. And here's my sources.
In stage 4 of cholesterol synthesis, the linear squalene molecule is converted to the 4-ring steroid nucleus. In this step, the relationship between the linear structure and the cyclic structure of sterols become clear. All sterols have the 4 fused rings that form the steroid nucleus with the hydroxyl group as C3. Squalene monooxygenase is the main enzyme in the final step of cholesterol synthesis. It is located in the endoplasmic reticulum and is conserved across all eukaryotes. Squalene monooxygenase contains the N-terminal, which contains the 3 distinct domains for the FAD binding domain. It also contains the substrate binding domain and a C-terminal helical membrane binding domain. The enzyme undergoes conformational shifts in the substrate binding domain. It also contains an inhibitor binding tunnel, which also acts as the squalene binding site. It adds 1 oxygen from O2 to the end of the squalene chain, forming an epoxide. This enzyme is another mixed function oxidase, where it uses NADPH to reduce the other oxygen atom of O2 to H2O. The double bonds of the product, squalene 2,3 epoxide, are positioned so that a concerted reaction can convert the linear squalene epoxide to a cyclic structure. A concerted reaction is a chemical reaction in which all bond breaking and bond making occurs in a single step. Oxidosqualene cyclase is then used to create the cyclic structure. In animal cells, this cyclization results in the formation of linosterol, which contains the 4-ring characteristic of the steroid nucleus. Linosterol is finally converted to cholesterol in a series of about 20 reactions that include the migration and removal of methyl groups. After these reactions, the end result is a cholesterol molecule.
So if we look at the process as a whole, what we'll find is that we have 3 A-CoA, which become mevalonate in step 1 and step 2. Those will become activated isoprenes, isomers, 2 isomers of that, at which point step 3 they will join together to form a squalene, and in step 4 that squalene will be transformed finally into a cholesterol, first becoming an epoxide, then a linosterol, and finally a cholesterol. And what we see here is the gradual transformation of a basic molecule A-CoA into the more complicated and necessary cholesterol. And we chose to specifically highlight an enzyme in each of these processes. So in step 1, we highlighted HMG-CoA reductase, which is the target of statins, which is the rate-limiting step for the whole process. In step 2, we looked at IPP isomerase, which actually produces the 2 squalene isomers that are needed in step 3. And in step 3, we looked at squalene synthase, which actually completes the process of making a squalene. And this is very important because this is the committed step of the process. At this point, the process only goes one way. It only goes towards making cholesterol, whereas in other steps, the product can be used for other processes. And finally, we looked at squalene monooxygenase, which forms a squalene epoxide, and the oxygen that it adds will later become the alcohol group found on cholesterol, which gives its very unique properties. In addition to all this, we also tried to show the chemical properties of each of the steps. So what does this substance do? What are its properties within the cytosol? And that really relates to what we highlighted, which would be the adding and removal of carboxyl groups, the adding or removal of phosphate groups, all in the service of making cholesterol. And finally, we also highlighted the energy requirements of this and found that there were 36 ATP required and 16 NADPH in order to join and reduce the products made from combining in total 18 A-CoA in order to form a cholesterol. And it's our hope that this was clear.